So now we're going to really get into the nitty-gritty of urine formation. We kind of did an overview, but now we're really going to get into it. So we're going to talk about the events for urine formation. We're going to talk about glomerular filtration rates and the forces that regulate net filtration pressure. We'll talk about that, the mechanisms regulating that. We'll talk about tubular secretion and reabsorption. We'll talk about what happens for those things in the PCT. We'll talk about what happens in the loop of Henle. All right, as well as the DCT, and we'll get into the different hormones and how they will affect the reabsorption and secretion within this renal tubule. So a lot that we're going to get into, but I'll kind of point out some of the really big key things I really want you to take away. All right, so the solute to excrete a solute, right, to kind of get rid of things, we have to filter things out of the blood. We have to maybe add in more, right? So glomerular filtration is like the initial filtering process. And then some things are going to be secreted later on throughout the nephron, and some things are going to be reabsorbed. So generally speaking, if things are filtered, this is like this is very general. There is no deciding factor in terms of what becomes part of the filtrate except for size and charge. When it comes to reabsorption, typically what we're doing with reabsorption is we're reabsorbing things that the body can still utilize. So things like glucose, which is a nutrient we can still use, that's something very useful. Something like sodium to help maintain things like blood volume, those things are still useful. We want to pull them back in. Secretion is going to be for things we don't need, right? It's going to be waste products, things we want to have left in the urine. And then by the time we get through all of these steps and we get to the very end, we're left with the urine to be excreted out. So let's go ahead and start with glomerular filtration. Now we're super highly vascular as a kidney because that makes sense. Our job is to filter the blood. It makes sense we would have a lot of blood vessels and that we would receive a lot of blood on a regular basis. So we get about a quarter, right, a fifth to a quarter of the cardiac output. That's going to let us filter about 180 liters of blood plasma every day. Obviously, you don't produce 180 liters of urine. Most of that gets reclaimed. You make about a couple liters, right? Now, when we do this filtering process, we're going to form what's called the filtrate. And the filtrate is going to compose of water and all the solutes that are present in the water, including ions, glucose, amino acids, waste like uric acid, urea, creatinine, with the exception of large proteins. So larger proteins, and medium-sized proteins, like proteins in general are really big. They're not going to be part of the filtrate. Same with blood cells, platelets, red blood cells, white blood cells. Those will also not be filtered out. So everything else is filtered. The only things we do not see in the filtrate in this this first step, the only things we do not see are cells and proteins. If we see those things, we know something's going wrong. This filtrate then will pass from the glomerulus and that capsule from the renal corpuscle into the proximal convoluted tubule. Now, how much we have going through, right? How much plasma gets to the kidneys, right, for any given amount of time is called the renal plasma flow. So basically, what's the volume of plasma that gets to the kidneys for a given unit of time? So we can go over over a minute, over a day, right? Usually we're talking about per minute. So we see that 80% of the plasma that flows through from the afferent to the efferent arteriole is not filtered, but 20% is filtered into the capsule. This is where we get to the term glomerular filtration rate, or GFR. This is basically how much filtrate is formed by both of the kidneys, so just the kidneys in general, each minute. We see that we're producing about 125 milliliters of filtrate per minute in a male, typically, and about 100 mils per minute in a female, typically. This is assuming things like good health. So if someone has something like kidney disease, we would not see this amount of filtrate being produced. But this is what we're looking at. What's the volume, right? The glomerular filtration rate. What's the volume of filtrate we produce every minute? And when it comes to the filtration process, right, we have kind of a few layers here. Let's go ahead and talk about these layers. So we have the capillary itself, the glomerulus, the capillary, which is made of a fenestrated capillary or fenestrated endothelial cells. We have a basement membrane surrounding that, and then we have the podocytes. And so these three are basically going to form three different barriers for filtrate to cross. So the glomerular capillary endothelial cells, they are heavily fenestrated, meaning we have these big pores, these openings that are large enough for a lot of components of the blood plasma to get through, but not so big that cells and major proteins can. The basement membrane, also called the basal lamina, is going to prevent these larger proteins. So we have the fenestrated endothelial cells, which will stop cells. The basement membrane, which will stop large proteins. And then the podocytes, the little gaps between the pedicels or the pedicles of the podocytes, have these filtration slits. These are going to filter even more, so they're gonna stop medium-sized proteins. So you see how every layer from the inside, as we move out, we're getting a little bit stricter in what we're letting through, right? We're getting a little bit stricter in what is able to get through. So the fenestrated endothelial cells they're the most open. They allow a lot through, but then the basement membrane stops a little bit more. And the pedicles of the podocytes, they stop a little bit more. We get a little bit more of like a sieve type effect. Now, the glomerular filtration rate is going to depend on the net filtration pressure, which this is very similar to what we saw when we saw the Starling's law of the capillaries, when we looked at how systemic capillaries will make that filtration reabsorption happen. Here, though, we're only looking at three forces. We're looking at the glomerular hydrostatic pressure, which is the pressure that the fluid inside the glomerulus is pushing out. So this is going to push fluid from the blood into the capsule. The capsular hydrostatic pressure, this is going to promote reabsorption. This is the hydrostatic pressure of the fluid in the capsule pushing back at the blood. So that would push fluid from the capsule into the blood. And then the glomerular colloid osmotic pressure, this is from the proteins that we find in the blood, like albumin, which will pull fluid from the capsule space into the blood. So we have one pressure that is pushing fluid from the blood into the capsule, and two pressures that are pushing from the capsule into the blood. But generally speaking, that glomerular hydrostatic pressure is higher than both of the other two combined. And so we can calculate this out. The net filtration pressure is going to be the glomerular hydrostatic pressure minus the Bowman's capsule pressure, which is the capsular hydrostatic pressure, minus the glomerular colloid osmotic pressure, right? Sometimes you'll see the capsular hydrostatic pressure and the glomerular colloid osmotic pressure added together and then subtracted from the glomerular hydrostatic pressure. It will give you the same number, regardless of how you do it. So what we see is that the glomerular hydrostatic pressure is usually about 10 millimeters of mercury higher than the other two, which means we have a positive number, and a positive number, as we learned when we looked at the capillaries, is filtration. This is promoting filtration. And we see that's normally about 10 millimeters of mercury on average. That's about normal. If things are too low, then that means we're not producing enough filtrate, which means waste products may be building up. If our number is too high, then it probably indicates that some of those plasma proteins have made their way into the capsule and are drawing fluid out, and that can lead to what's called systemic edema. So we don't like, the kidneys are picky. They like things to be consistent. They don't like major changes in blood flow, because if the blood flow is too high, substances go through too quickly and we don't reabsorb them. We're basically, things are pushed along too fast, and the things that are still useful do not end up back in the bloodstream. We lose important, useful materials. And if it's too low, then we are reabsorbing a lot of stuff, including waste products. And we don't want to, the whole point of the kidney is to get rid of these waste products, not to reabsorb them. And we want to get them out. We want to excrete them. And so if blood flow is too low, some of those waste products are going to be absorbed too much, and we're not going to be properly getting rid of them. So how do we control this? This is going to be controlled via the vasoconstriction or dilation of those afferent arterioles. Basically, we can use those arterioles to control how much blood flows into the kidneys at any given moment, right, into those nephrons. Some of this is going to be through intrinsic autoregulatory mechanisms so that we can control ourselves within the kidneys. Some will be through extrinsic autonomic nervous system regulation, so that sympathetic innervation we talked about earlier. And then the extrinsic hormonal regulation, right, will also kind of tie into this as well. So a few different ways for us to control how much blood flows through the kidneys at any given moment. And we can, depending on what's going on, we can, let's say we don't have enough blood flow going through there, maybe autonomic nervous system kicks in, or maybe a hormone kicks in. Something to try to keep it relatively constant. We want to try to keep it relatively constant. So let's go ahead and talk about the hormones that will influence the glomerular filtration rate. One is angiotensin II. Angiotensin II is part of that renin-angiotensin-aldosterone pathway. Basically, what this is going to do is angiotensin is going to help promote higher blood pressure. And to do that means we're going to be constricting arterioles. It's going to help us increase pressure. So when we constrict the afferent arterioles, that's going to lower our glomerular filtration rate, which overall will lower our urine output. So we'll get into the RAA pathway a little bit more in a little bit, but that's all right. ANP, or atrial natriuretic peptide, sometimes called atrial natriuretic hormone, or ANH, this is going to increase urine output, because this is going to do the opposite. But when blood pressure or blood volume is too high, it's going to tell the cell, it's going to influence sodium reabsorption negatively. It's going to cause us to not reabsorb as much sodium. But it's also going to relax these mesangial cells at the JGA. This is going to increase surface area, which increases the flow rate, which increases urine output. It's also going to suppress the RAA pathway. Since we are trying to lower blood pressure, we don't need as much angiotensin then, since angiotensin, the RAA pathway is involved in increasing blood pressure. So let's go ahead and talk about RAA, the renin-angiotensin-aldosterone system. And so for this one, we're just going to I'm just going to pick a place to start. We're going to start with angiotensinogen right over on this side, and we'll work our way across, kind of talk about what it does. So the big thing, let's say, is we have some sort of decrease in renal perfusion, right? We're going to release renin. This means that blood pressure is too low, right? We need to increase blood pressure, increase blood volume, right? So we are detecting that in the kidneys, and we are going to release renin. The liver is making this kind of precursor, this enzyme precursor called angiotensinogen. Renin will activate angiotensinogen to angiotensin 1. The lungs then will also help with this. They're going to produce the ACE, which is angiotensin converting enzyme. Right, that's what that stands for, which will then take angiotensin 1 and convert it to angiotensin 2. Angiotensin 2 is then going to have all of these different effects. So let's go down the list. It's going to increase sympathetic activity. It's going to help us reabsorb sodium and chloride and get rid of potassium, which means if we're pulling in the sodium, we're also going to be retaining water. We're going to secrete aldosterone. Right, aldosterone is going to help with this, with the same function. We are going to constrict the arterioles, which is going to increase blood pressure. That's why we angiotensin, right? Angio, we think blood and whatnot, right? Cardiovascular, tensin, we think pressure. So we're going to increase blood pressure. We're also going to stimulate the pituitary to release ADH. That ADH is going to help promote the reabsorption of water. So you can see how all of the, how all of this is going to help all together, right? We're going to be basically increasing blood pressure, which is going to drop our glomerular filtration rate, so less urine output. Now we can auto-regulate, right? So even when we have like major changes in blood pressure, we can auto-regulate as well. So maybe something's going on, right? That is causing our blood pressure to spike. And so we don't want that high blood pressure to, vastly change how much blood is flowing through. And so if the systemic blood pressure is really high, afferent arterioles will constrict to lower the filtration pressure. If the blood pressure is really low, afferent arterioles relax so that more can flow through. So increase that pressure, increase that GFR, which increases our urine output. And there are two ways that we can auto-regulate. Myogenic mechanisms, which is basically like there is a stretching of the arterial wall, which induces muscle contraction, right? So if we have higher blood pressure, it's going to push on the arterial walls more. And as we push on those arterial walls more, that's going to stimulate the smooth muscle in that arteriole to clamp down, to contract. The tubuloglomerular feedback, this is where we get in that juxtaglomerular apparatus, right, where the macula densa is going to inhibit the release of nitric oxide, which is going to cause vasoconstriction and higher blood pressure. Or the macula densa releases renin because blood pressure is too low, right? Which then activates our RAA system, which will then promote increasing blood pressure. So those are a couple ways that we can auto-regulate where the kidney itself can influence blood pressure, blood flow, and all that to maintain a relatively constant blood flow through the kidney itself. When it comes to innervation, right, the sympathetic nervous system is really what's kind of keyed in here. They're releasing norepinephrine, which is going to go onto those alpha-1 adrenergic receptors. This is going to cause vasoconstriction, right? When these afferent arterioles vasoconstrict, we drop that GFR, we reduce that urine output, right? And this is so that we can divert blood to other tissues that are necessary for immediate survival. So things like your heart, things like your skeletal muscles, things that your lungs, things that are going to help promote immediate survival, immediate safety. Now when it comes to tubular secretion and reabsorption, this is coming back down to, well, a lot of stuff that entered in as the filtrate early on is still really useful. There's a lot of water, there's a lot of useful solutes that we still want. And so reabsorption is going to be our process of kind of selectively choosing which of those components we bring back into the bloodstream, back into the body. Tubular secretion is for things that maybe didn't make it into the initial filtrate, but we do want to get rid of. So this is going to help when it comes to maintaining things like pH as well as getting rid of other waste products. So let's go ahead and talk about how things are reabsorbed. What can we do to reabsorb things into either into the interstitium, which is just into the interstitial space, or into the peritubular capillary that runs all around the nephron. We have paracellular reabsorption. This is passive, so this just means things are diffusing between the cells. So for example, up here we see this sodium ion just passing between the cells. This is passive. We don't need any energy input for this. Or transcellular reabsorption, where the cells forming the walls of the tubule will actually take that substance in and then using ATP transport it through the cell to the other side and out. So here we see our sodium doing the same thing, where we've pulled it into this cell that is in the wall, right, that forms part of the wall of the nephron, and then we use ATP to pump it back out so that it can enter into something like a capillary. Since it's active transport, we do require the use of ATP. So we have a couple options, and it depends on what we are reabsorbing and where we are in the nephron, and some other factors as well. But we're going to use one of these two methods. So active transport, the majority of solute reabsorption is going to use active transport using using sodium gradients. So we have sodium transporters, basically, that will help promote the transcellular reabsorption of things like glucose, of like the amino acids, of bicarbonate, of sodium, of potassium, of chloride, of calcium, of magnesium, of phosphates. Right, these are going to require some energy input. Normally, 100% of the glucose, the amino acids, the lactic acids, vitamins, right, things of that are reabsorbed in that first part of the proximal convoluted tubule, and we're doing that by these sodium symporters. Now diffusion, right, sodium that's in the interstitium space from active transport is then going to help drive the diffusion of potassium, calcium, magnesium, and chloride using the paracellular route. So because we have all this sodium in the interstitial space, using these symporters, we can then use it to pull other ions with it. We also, because we have all this sodium, we can create an osmotic gradient, which basically promotes osmosis, right? Water is going to be pulled by that high sodium concentration, so water will just pass through those cells or in between those cells passively. So let's go ahead and talk about water a little bit more. So the diffusion of water, this is a major job because we have so much water in that filtrate to start with, and we don't want that much to end up in the urine. We'd be urinating all the time if that were the case. So we need to reabsorb it. So let's go ahead and talk about how. We have the obligatory water reabsorption in the tubule. This is going to be most of it, about 85%, where water is following the solutes. Right, we have aquaporin transporters, and water is following the solute concentration just through osmosis. The facultative water reabsorption, this is the last bit. This is what's happening in the collecting duct. And this is where we're using aquaporin 2 transporters, and this is going to be through ADH. So ADH is going to promote these aquaporin 2 transporters from being put on the surface of the cells. If ADH is not there, we don't see all of these aquaporin 2 transporters. So ADH regulates the aquaporin 2 transporters in the collecting duct area. So now let's go ahead and talk about what's happening in the proximal convoluted tubule. If we see this is the first part that's going to be getting all of this filtrate. What we're going to be getting, we're going to reclaim, so we're going to reabsorb back into the bloodstream a bunch of water, right? 65% of it. We're going to get a lot of sodium, right? A lot of potassium, all the glucose, and all the amino acids. And these are key. And if you notice, right, why so much water and sodium and all that is that water and that sodium, first of all, is a symporter. So we're going to use that sodium to reabsorb things like the glucose and amino acids and potassium. And it's also going to help pull that water with it. We're also going to get some of the chloride ions, some of the bicarbonate, urea, which is one of our nitrogenous waste products, and calcium, magnesium, right? Those are going to use diffusion. So you notice a good chunk of these are using active transports like the sodium, the glucose, the amino acids. Others are using diffusion. We're also going to secrete things like our hydrogen ions, our ammonia, urea, and creatinine. So secretion, remember, reabsorption. So we're going to reabsorb some of the urea, some of the waste products, but we're also going to secrete some back in. And so everything we see there under secretion, that is going to be pushed into the urine, from the blood into the urine. At this point, we're still going to be isotonic to blood, so the pH is not any different from what the blood is at this point. When it comes to the secretion of wastes, right, those hydrogen ions, the urea, the ammonia, we have sodium hydrogen antiporters. Basically, we reabsorb sodium and bicarbonate, and by doing that, we're pushing the protons into the tubule. This is going to help with pH regulation because that bicarbonate is a buffer. We're reabsorbing something that has a negative charge when secreting something with a positive charge. We'll also secrete urea and ammonium via antiporters. So once we're in the tubules, right, we have that urea and that hydrogen ion bind to form that ammonia. Now with glucose, we're using a glucose and a sodium co-transporter, meaning we're basically going to be, glucose is going to kind of hitch a ride with sodium as sodium is brought in. If there's too much glucose in the filtrate, though, there's no way that we can reabsorb it all because the carriers are totally saturated. So, like, this is where, like, if your blood glucose level is really, really high, there's just no way for you to reclaim all this glucose from the filtrate, and so you do see some glucose end up in the urine. This is called glycosuria, and this is a sign of diabetes mellitus. So that would be a sign that the blood glucose levels are just so high that there's so much glucose in the filtrate that we just don't even have the time or the ability to reabsorb it all.